Oren. I'm presenting a microsphere-based delivery system for the targeted administration of the antifibrotic peptide SDKP, bit of a mouthful. So I did this research under Professor Joachim Kohn in the New Jersey Center for Biomaterials. And I'll just start off with some background on the disease that this is aiming to treat. So your kidney filters toxins and excess ions out of your bloodstream. And the functional unit of the kidney is the nephron, showing that black rectangle there. And within the nephron, the bulk of actual blood filtration occurs in a tightly wound structure of blood vessels known as the glomerulus. Now, the glomerulus is held in place by a complex structure of extracellular matrix, or ECM, proteins. And this extracellular matrix is produced and assembled by these purple mesangial cells placed throughout the glomerulus. Now, in diabetic patients, the blood can become very hyperglycemic, and under hyperglycemic conditions, the mesangial cells lose control of their ECM production. And so they start expanding into space that was previously occupied by blood vessels. And this restricts the blood flow, and if this continues to an extreme extent, the body will actually bypass blood flow around the glomerulus, and so no actual filtration of the blood will occur. And this condition is known as diabetic nephropathy, and it can lead to a whole host of medical complications, including fluid retention that may require limb amputation, blindness, and most importantly, the often deadly kidney failure. So there do exist some treatments for diabetic nephropathy: kidney transplant, dialysis, and ACE inhibitors. So first, kidney transplants. Kidneys are some of the most sought after transplant organs in the world. And the need for kidney transplants, as you can see in the graph on your left here, far outweighs the availability of transplantable kidneys. And this gap between the need and the supply is growing rapidly and steadily over time. Part of the problem here is that in order to get a kidney transplant, you have to be compatible with the blood type of the kidney, and this results in an average of a 3 to 5 year waiting period for a kidney transplant. And on top of this, around 14% of patients who receive a kidney transplant experience kidney failure again within the first 5 years after the transplantation. So another option, dialysis. Dialysis involves going to a hospital, getting hooked up to a big machine that takes your blood, filters it for you, puts it back in your body. And patients on dialysis, on average, need to go to the hospital for around four times per week for a 3-hour session each time, around 12 hours a week. They have to implement dietary restrictions, and even on dialysis, patients have about a 5 to 10 year life expectancy after beginning dialysis. So the pharmaceutical approach to treating diabetic nephropathy is using ACE inhibitors. Now, ACE inhibitors, they target the epithelial cells around blood vessels and relax them, allowing the blood vessel to expand a little bit and allow for a little bit more blood flow. But the problem here is this ECM expansion is systemic and continuous. And so eventually the ECM catches up, and there's only so far you can relax the blood vessels. So this may delay the need for kidney transplant or dialysis, but it does not eliminate it. So my proposed solution is the delivery of a drug that actually stops the production of extracellular matrix proteins encased in a biodegradable polymer sphere with a targeting mechanism mounted on the outside of the sphere that targets specifically kidney proteins. So as the sphere travels through the bloodstream, the targeting mechanism will attach to kidney-specific proteins and anchor the sphere in place while it degrades over a period of about 1 to 2 months, releasing the drug. So the first step in my experimentation involved selecting an antifibrotic drug. So after an extensive literature review, I selected the peptide sequence SDKP, which has been shown to downregulate extracellular matrix production. So after synthesizing the drug, I cultured it with human dermal fibroblasts for 2 days, just to see if the drug is killing the cells rather than treating them. And I used immunofluorescent staining methods to show here that there is no significant increase in cell death in cells exposed to SDKP versus those that aren't. The next step was to determine the effectiveness. So I repeated this experiment, but instead of staining for live cells versus dead cells, I stained for the extracellular matrix protein marker, fibronectin. And you can see the difference. The difference is subtle, but you can see it's slightly darker in the cells that were exposed to SDKP over that period of 2 days, showing some preliminary results that the SDKP is successful in limiting the production of fibronectin by these cells. So now we have the drug, but we're not quite done yet for an important reason. The drug limits the production of extracellular matrix. And extracellular matrix is vital for a lot of biological functions, including cell signaling, cell attachment, tissue structure. It holds your organs in place, keeps your meat on your bones. If you were to just inject this drug into the bloodstream and let it have free reign over wherever it targets your cells, you'd essentially fall apart. Trying to rid yourself of diabetic nephropathy by injecting an antifibrotic drug into your bloodstream would be like trying to rid yourself of a tick that's hanging onto you by jumping into a volcano. Not a great idea. So, solution for that, limit its release in a biodegradable polymer sphere with slow release. So the solution here is to just insert it into a polymer sphere, but this still doesn't limit where the drug goes. It just delays its release. So the third step in the process here is to mount the targeting mechanism on the outside so that the drug is only delivered to the kidney. So in this process, we now have the drug. Next step is loading the drug into the sphere. So traditional microsphere drug delivery methods involve polymers that have outer chains that are hydrophilic or water-loving, and inner segments that are hydrophobic or water-fearing. And when immersed in water, these spheres spontaneously form microspheres with a hydrophilic outer layer and a hydrophobic core. And this is great for transporting hydrophobic drugs that also want to stay out of the water. But unfortunately, SDKP is a little bit too hydrophilic and would be likely to diffuse out of the hydrophobic core, out of the sphere, before it even degrades. So to counteract this problem, I employed emulsion techniques that create a sphere shell out of hydrophilic polymer that contain another hydrophilic medium on the inside so that it can contain hydrophilic drug while still separating it from the hydrophilic environment of the bloodstream. So here are some results of some attempted drug loading that I carried out. So this is using dynamic light scattering to analyze the size of the spheres. So I hypothesized that given a constant method of creating the spheres with drug loaded inside will show a larger diameter just because of steric hindrance. And that is what I was able to show here. You can see a steady increase from sphere size with no drug loaded to 5% weight drug loading to 10% weight drug loading, suggesting that I was successful in loading the drug into the sphere. So we got the drug in the sphere. Next step is attaching the targeting mechanism. So when experimenting with targeting mechanisms, the microspheres themselves are very time-consuming and expensive to produce. So I just went for proof of concept experiments here. So instead of attaching to the spheres themselves, I used polymer grains, just like a fine powder, to simulate the surface chemistry of the polymer. And so I selected another peptide sequence, CLP-VSC, which has been shown to bind specifically to kidney proteins. And this is not to be confused with the peptide SDKP. Both peptides, one is the drug, one is the targeting mechanism. So this is the targeting peptide. So the CLP-VSC peptide has a carboxylic acid side chain, and as does the polymer itself. And so theoretically, these carboxylic acid side chains could be cross-linked together if facilitated properly, such as through an EDC-NHS reaction, which I used here. So I attempted attachment of the peptide to the polymer grains using EDC-NHS reaction chemistries, and then ran an NMR on the polymer with peptide attached. And so this is polymer with peptide here, and just comparing that to just the polymer, they're looking kind of similar. So there are a few differences here, but not really enough to definitively say, yes, I attached the peptide. And this doesn't necessarily mean the peptide wasn't attached. There could be several reasons for it not showing up well in the NMR. For one thing, these polymer grains have a much higher mass ratio to the, have a very large mass ratio to the peptide I'm attaching to the outside, because it's only attached to the surface. So all of these gigantic polymer peaks really overshadow any results coming from the peptide itself. Also, the spectrum, the NMR spectrum of the peptide overlaps significantly with the NMR spectrum of the polymer, and so a lot of peaks that would show up for just the peptide get swallowed up by the polymer peaks. So I went a slightly different route to just prove the idea of using this EDC-NHS reaction chemistry to cross-link to the polymer. So to show it visually, I tried using a dye, safranin O, which also exhibits the same carboxylic acid side chain and should be able to attach in the same way. And I used the same attachment experiment. And you can see on your right here, this is polymer with dye, and on your right, this is polymer with dye and the facilitation of the cross-linking. And they don't really show a whole lot of difference there either. And this is likely because the safranin O molecule itself is a lot smaller than the peptide, and it might be small enough to actually soak into the polymer grains so that the grains become colored even if it didn't attach. So this didn't really provide any proof of attachment either. So on to the third attempt at proving attachment. I synthesized a new peptide, just to simulate the real peptide we want here. A fluorescently tagged chain of lysine and glycine amino acids. And I attempted to attach it to polymer grains using the same methods, and then just image the fluorescence. And here, now we finally see a clear difference. So you can see an increase in fluorescence from just the polymer plus the peptide, where some may have soaked in, to polymer plus the peptide plus attachment. There is some fluorescence in all of these because there is some background fluorescence from the polymer that overlaps with the peptide fluorescence, but it's the jump in fluorescence from these two to this last picture here that show that something was actually attached. And I also, just to use an analytical approach as well as the visual, I ran these samples through a fluorescence plate reader to show relative fluorescence values, and you see that same increase coming from the machine. So I've developed a drug that can be used for this purpose. I loaded it into a sphere successfully, and I exhibited proof of concept for attaching a targeting mechanism to the outside of the sphere. Next steps for this project include the optimization of the microsphere size and loading to maximize the drug to polymer ratio, running in vitro experimentation with loaded spheres to ensure that loaded spheres don't exhibit cytotoxicity and still exhibit the same effectiveness, and eventually moving on to animal models to prove the effectiveness of the targeting mechanism as well as the drug effectiveness in vivo. And with that, thank you very much.
Does anyone have any questions at this point?
Yeah, Caitlin.
Can you go back to the slide where you showed like your staining with the cells, like earlier on? I just had a question. you see an increase in actin?
Great question, not a clue.
Okay.
We stained for actin expecting it to come out around the same.
Mhm.
We didn't see any changes in cell behavior that might indicate that the actin was upregulated.
Mhm.
So we're not entirely sure how to explain that, but I included it in the slide here because, it's something that I thought might mean something. We're not entirely sure yet.
Okay. Do you know what stain you used?
Rhodamine phalloidin, I want to say.
Okay, because that stains